Let's take a look here at the bone marrow. You can see that you have some stem cells here and over here. You also have some mature cells and some that are on their way of maturing. In general, you have some pathways right here. You're starting off with a stem cell, and then the maturation process then leads to the mature blood cell. So here in this case, we're making red blood cells. And so we're starting here out with the stem cell, then we have erythroblasts, and then the last stage will be the reticulocyte, and it kind of shoots out its nucleus right there, because red blood cells, they don't need to have a nucleus, and that makes them more flexible and gives more space for hemoglobin. So then they are entering the bloodstream right here. Those are mature red blood cells. And yeah, in this picture, you're going to have also neutrophil maturation. Again, we're starting out with the stem cell right here, and then going through various blast stages until you have a mature neutrophil right here that enters then the bloodstream. Now focusing in on those red blood cells, they have this sucked in center because they don't have a nucleus anymore. And it makes them more flexible. It also helps them to pack up with more hemoglobin. And of course, that's their function. The disadvantage is that the lifespan is limited. Once the hemoglobin is worn out, and the cell, it can't make more because it doesn't have a nucleus, so then these cells will have to be recycled. So they have a lifespan of about 100 to 120 days approximately. Sometimes people study red blood cells because it's sort of a simplified cell surface right here, and people like to study the cytoskeleton, these connections right here with the cytoskeleton. That's kind of interesting here. Here are different shapes of red blood cells. So over here, we have a red blood cell that looks dehydrated. So it was placed in a hypertonic solution, has lost some water. Now you can see how it's all spiky and dehydrated. Here, all these spikes. Here, this red blood cell was placed in a hypotonic medium. And well, luckily that it hasn't burst yet, because normally red blood cells burst very quickly when you place them, for example, in distilled water. But here, just maybe slightly hypotonic solution, the red blood cell swells up, and there's always a danger it might burst. Here's some abnormal hemoglobin that's causing the red blood cells to deform. You can see this sickle cell red blood cell that looks like this half moon shape, has this sickle cell. I guess fingernail clippings is another analogy. And the morphology in general can provide clues to the presence of disease. There are different values that people measure. One of them is the MCV. MCV stands for mean corpuscular volume, with corpuscle meaning the cell itself. So it measures the size of the red blood cells, and it could be either microcytes, cells that are too small, or macrocytes, cells that are too big. And so this mean or average red cell volume, that's the mean corpuscular volume, and this MCV is measured on every general blood screen that people do, like a complete blood count, a CBC. That's a typical kind of measurement. So here is your hemoglobin molecule. It has 4 subunits, 2 alpha and 2 beta subunits. So we have here in lighter purple the 2 alpha chains. Here's the other one. Then in a little darker purple, the beta chains 1 and 2. And then in each one of these chains, you're going to have this heme group. This here overall is called a porphyrin ring, and it's a fairly complex organic structure. The important thing is this iron, the central iron. And this iron here is the place where the oxygen is being attracted to. And it's not a super strong attraction, so it can come off there again. And it's a very fine-tuned balance where the oxygen is sort of being parked onto this iron, but it can also come off of this heme group with the iron in the center. Now here, the hemoglobin and the iron, a lot of people don't have enough iron, and then they end up with something called iron deficiency anemia, because you must have iron. If you don't have iron, you can't make these heme groups, and without heme groups, your hemoglobin doesn't work. So typically you want to recycle as much as possible your iron. You also need to make sure that you get enough iron in your diet, and you're supposed to absorb the iron from the blood, from your intestines, from your digestive tract, and then you will be this transferrin. That's a protein that will transport iron in the plasma of your blood, and it transports it in the plasma to the bone marrow where you're making all the red blood cells. So it's used for the heme group. The iron needs to be in the heme group for the hemoglobin and for the complete red blood cell synthesis. So then once we have hemoglobin, then we can fill it up into these red blood cells. The recycling, the way it works, so these red blood cells, they live about 120 days in the blood. And then you want to recycle them, and that's done in the spleen, for example. And it will try to recycle as much as possible the iron. Now, but there will also be some byproducts from the hemoglobin, and these are colored, pigmented kind of molecules. So the hemoglobin will be converted to bilirubin, and bilirubin, that then these pigmented metabolites, they're being excreted in the urine and in the fecal material. And so that's basically the reason why your urine is yellow and the fecal material is all brown. So here, the kidneys will take care of the water-soluble bilirubin metabolites and excreted via the urine, and then in the other liver, these other bilirubin metabolites, they will be excreted in the fecal material right here. So bilirubin metabolites in the fecal material. Here's sort of your summary for the red blood cells. They live about 120 days. The hemoglobin components are recycled as much as possible, especially the iron needs to be recycled. And then we do have some waste products that get excreted via kidneys or the fecal material. Also, once you're taking this hemoglobin protein apart, you want to recycle the amino acids and incorporate them into new proteins. You want to recycle as much as possible the iron and use it for new heme groups. And then the remnants of the heme groups are converted to bilirubin, excreted as bile, and via the kidneys as well. Somebody that has, or a baby that was maybe born prematurely, that doesn't have the liver function, hasn't picked up yet, that they might end up with jaundice, which results from elevated bilirubin levels. Also, people that have hepatitis, they would end up with jaundice because the liver can't process all of the bilirubin. Now let's take a look at some causes of anemia. And here, of course, the most intuitive form of anemia would be that you've lost a lot of blood. You are bleeding, and so that would be simply blood loss right here due to an injury, perhaps. Then there are various forms of hemolytic anemia. Hemolytic anemia could be hereditary. The most often heard genetic disease associated with hemolysis would be sickle cell anemia. That is when your red blood cells are deformed and they rupture all the time. And then there are some acquired forms of anemia that would be due to parasitic infections such as malaria. Some drugs also lead to anemia, and then some autoimmune reactions. Now, decreased red blood cell reaction could be due to aplastic anemia, which is usually due to radiation. Good example for that would be on people that were too close to the Chernobyl reactor when it exploded. A lot of them, if they didn't die from too much radiation, but a lot of them ended up with aplastic anemia. And then sometimes inadequate dietary intake of essential nutrients. Most common here would be the iron deficiency, not enough iron in the diet. Also folic acid could be a deficiency. And then something that strict vegetarians should know is vitamin B12. You need to get that from animal sources or from a dietary supplement. But if you're strict vegetarian, you might end up lacking vitamin B12, and you do need vitamin B12 to make red blood cells. And so sometimes people also have a problem with the intrinsic factor, and then they will not be able to absorb the vitamin B12. Now here, moving on to megakaryocytes. They kind of look kind of rough on the outside, and they constantly chip off these little pieces, these platelets, chipped off pieces from the megakaryocyte, and you do need those in order to have blood clotting. So down here you have an inactive platelet, and over here an activated platelet. And this slide we had before, I'm not sure why this is in here again, but let's move on to hemostasis and tissue repair. So here you have the vasoconstriction. That's the first thing that happens once you have an injury. And then there's a whole bunch of different things that set in. I don't find this particular flowchart very helpful. It's a little bit confusing, I think. But there are a few things that will come in the following slide that you should know. So you do need this thrombin, and you need to convert fibrinogen to fibrin. That's very important. And then you form this clot, and then you're going to have this plug that seals a wound or some sort of a cut. Here would be the platelet plug formation. You can see right here how there was an injury, and now these platelets, they get stuck in a mesh of fibrin, and then these platelets, they will seal this open area and prevent more blood loss here from this blood vessel. Okay, here is the laundry list of factors that are involved in platelet function. There's a lot of them, and I don't have much intention of going over these here. Serotonin is maybe one thing that you should know about. There's also platelet-derived growth factor, but let's take a look at the clotting cascade, and maybe that will all tie it in for you. So basically, you're going to have three pathways that will have to converge. We have the intrinsic pathway, the extrinsic pathway, and the common pathway. So basically, you're starting, sort of dominoes start falling, a whole cascade of events that's going down this way, and another cascade of events going down this way. Eventually, they converge into the common pathway. Now, here is the thing. You need to have active factor 10. And for that, you need some calcium. Once you have active factor 10, you can convert prothrombin to thrombin. Once you have thrombin, you will convert fibrinogen to fibrin, and then the fibrin forms this mesh that causes these platelets to get stuck in there, and you form a clot. So one more time, you have intrinsic and extrinsic pathways. They converge to the common pathway. You need to have calcium. You also need vitamin K. I don't see this right here right now, but you do need to have vitamin K. And then the most important part here would be the conversion of prothrombin to thrombin. Once you have thrombin, it will convert fibrinogen to fibrin, and once you have the sticky mesh of fibrin fibers, then the platelets will get stuck there, and that will be your clot. So here, the most important part of the clotting sequence here in one slide, and I do agree with this slide. This is very nicely done. So basically, you need to convert prothrombin to thrombin. Once you have thrombin, then, right here. So the thrombin will convert the fibrinogen to fibrin. And once you have this mesh of fibrin fibers, then your platelets will get stuck in there. So that is the most important part. So here would be a nice picture. There is this mesh of fibrin, sticky fibers, and a bunch of platelets that get stuck there. Also, red blood cells get stuck in this mesh of fibrin, and they will seal the cut or the wound, and then you preventing further blood loss. So here, another list of these chemical factors that are involved with all of this. And you don't need all of this. You don't need to memorize this whole table here. But you should know about some essential factors that are important. So here, the prothrombin is important. The fibrinogen is important for the conversion of to fibrin. You need calcium. You need also vitamin K. These are essential factors that you must have in order to be able to clot. Here are some endogenous factors involved in anticoagulation. There's not an awful lot that you need to know here, just maybe the antithrombin and the prostacyclin, but of course you can take a longer look at this table. And here at the end, anticoagulants, those are important because clinically, because you want to prevent clotting, especially to prevent heart attacks. And here very important anticoagulants are heparin and antithrombin, protein C.